different picture. And also the scientific community, their role become more popular now. So with the COVID, people are looking at scientists finding solutions. Politicians are behind scientists, which is a great news. I think a good time for science now.
That could be seen to be one of the benefits of the COVID crisis, was that you heard politicians referring to the science so frequently for the basis of their decision making.
Exactly, yeah. And another thing which is sort of getting to this is a bit of hope, and I was talking to Shahid earlier. In the UK now, the DEFRA has done consultation with the community in terms of lifting the ban on GMO crops, or I would say species. So, but it's still they haven't finalized it, but the mood is pretty good. So we will be looking at a formal, I would say, outcome in next 3 months, which is a great news.
Yes, that would be interesting to hear what the UK does. They still have to export to the EU. I think that's the main problem.
Yes, that's the thing. You have already seen recent crisis with the vaccine as well. So it's becoming a bit of a, I would say, tit for tat thing.
Yes.
Khalid, I think we should finally start the program now.
Okay.
It's time now.
Exactly 1:30. Yep.
Okay. Thank you so much. Bismillahirrahmanirrahim. So my name is Dr. Khalid Mahmood. I'm today's host for the event. I would like to welcome all of participants from Pakistan and around the world, speakers and participants for sparing time to this event. The purpose of the event is basically developing a understanding and consultation with the stakeholders around where the Pakistan should go in terms of addressing the issues faced by climate change. And there is an emergency that we need to address to address food security in the country. And the government is very supportive. And the event is basically a follow-up of the discussions and I would say meetings happening over the last, I would say, 3 years now. The first consultation happened back in 2019 with Comstech and with the NIPG and Qaeda University and other partners in Pakistan, and with the government departments as well. So today we'll be giving more insight to what are the challenges that the food is facing, both coming from livestock and from crops, and where the opportunities are through modern genetics. And I mentioned earlier to my colleagues that I sit in the UK. I work for one of the large institutes in the world, one of the oldest institute, I would say. It's called Rothamsted Research. And we have some experiments that go back to 1843. And we've been doing a lot of work on wheat in terms of aphid-resistant wheat and some other interesting crops as well, because we need to produce more food per unit of land and per drop of water as well. So this is going to be a challenge. And when the climate change is happening, and when we see the rising temperatures, so all these things add up into a bit of a perfect storm. So there was actually a big study done back in the UK, 2008. Our chief scientist, Sir John Bennington, he predicted it's a perfect storm situation now in the world, where the resources are limited and the crisis coming from invasive species is high and high. And the food we're eating it in missing those nutrients. So essential nutrients like iron and zinc, they're deficient. And today's, I just see we have lined up speakers like Professor Buttasab, who will be explaining the challenges which are faced in the nutrition in Pakistan. Without taking much time, I would like to but I think just add a bit more, because this consultation happened in 2019, then follow-up meeting happened. I also represent UpSign. UpSign is a UK-Pakistan Science Innovation Network, which is linking scientists from all over the world with Pakistan. We had a huge event back in last March before COVID hit in Islamabad with development workshops. The idea was, how do we address the food security and this whole nutrition challenge through scientific innovation? So before going into more details, I would like to invite Professor Dr. Shahid Mansoor Saab, who's our, I would say, the distinguished professor and our fellow of the Pakistan Academy of Sciences. He's director of Pakistan's top-class institute, National Institute of Biotechnology and Genetic Engineering, which is based in Faisalabad. You might all already have heard about them. So I'll just give the floor to them and to just explain about the event today. Over to you, Professor Dr. Shahid Mansoor Saab. Dr. Shahid, please unmute yourself.
I'm saying that, can you hear me?
Yes, now we can hear you.
Okay, so thank you very much to everyone for joining us in this event on a very short notice. And I must thank in advance all the speakers for their, response to my call. I'll just tell that, we started this first event in 2019, and, as Khalid has already explained. So what we thought is that, we are facing this challenge of food security. Everybody is talking about this. And so I will go into some of the details that, that we need to produce more food from per unit area. Now, when you come to the Pakistan, then there are very interesting facts like, we are facing the problem of population, water, climate change, land, pest and diseases, salinity, and water logging. And as probably Dr. Mark Tester will tell us that, we are overusing our groundwater, and this is another challenge. With the green revolution, we were able to have self-sufficiency in cereals, sugar, fruits, vegetable, but we are net importer of many crops, and then additionally we have a very large burden of nutritional security because around 50% of our population suffer from nutritional deficiency. Then there are multiple ways of looking at the nutritional deficiency. One is management. That means that you can build more dams, have more land under cultivation, or you can work for genetic gain, which means that there are many historically there are many success stories not only in crop but also in milk, meat, and eggs. So we have some of the major progress made because of the genetic gain. But the problem that all the scientific community has realized is that the rate of genetic gain is slow. So we need new technologies to come up with the challenge. So as I mentioned earlier, that we have benefited from green revolution. So we need another revolution. We call it gene technology, and then there are multiple technologies that are to increase the genetic gain. One of these is genome editing, which has become, it is recognition of the importance of this gene that, this year's Nobel Prize went to the two ladies who actually discovered this technology. And so I'm not going into the detail that how it is done. Is it allows you to give to do very precise editing where you can either add or remove one or few bases, or you can replace one base with another base or few bases. And this was possible because of some very interesting progress which has been made in CRISPR technology. There's another very important development which has made in the genome sequencing technologies, and the cost of genome sequencing has gone down. Amazing technological advancement has been made, and which has allowed to sequence all the genomes. So there are now thousands of genomes available, human genome, as well as 30,000 plus rice genome. So that means that we have got a huge opportunity to use this genomic data, which is now freely available in public databases. There's another very interesting work done by one of our friend, Lee Hickey. And what he does is that, he played with lights and combination of lights, and, because in breeding technologies, one of the limiting factor is that generation advancement. So by using a right kind of a combination of light, he was able to establish what we call as speed breeding. A speed breeding is just a normal breeding. What it does is that, you know, it allows you to have 5 or 6 generation, for example, of wheat instead of 1 or 2, and that is a very big success that allows you to have a rapid genetic gain in major crops. Now, what we have done over the year is that, you know, because of the consultation process that we started a few years back, this was both formal and informal. You know, there was a consultation with Professor Ataur Rahman, Professor Iqbal Chaudhry, and others. And then we thought that, you know, we should have multiple platforms available. And we thought that, you know, if we have one in Faisalabad, which is located in central Pakistan, then one in Karachi or Sindh, which can cater the need of genomics and genomic selection in southern part of the country like Balochistan and Sindh, and then another facility in Islamabad or Mari. But now we think that, you know, we have now 3 partners which have over the year. So I'm very glad that, you know, we have Awais. I met Awais at CIMMYT, China, and I found him that very enthusiastic, very knowledgeable person. So we have developed this facilities, a platform that, you know, we will have 3 facilities. So with this, I thank all the audience for attending this seminar. And now I will invite Professor Iqbal Chaudhry, who is one of the leading soul behind all the success and development of this model. And he is also leading the Jamilur Rahman Institute of Genomics. So over to Professor Iqbal Saab. So Professor Iqbal Saab is the director and coordinator general of Comstech in Islamabad. Dr. Saab, please speak.
Bismillahirrahmanirrahim. Assalamu alaikum, and good morning to our colleagues in UK and in Canada. Thank you very much, Dr. Shahid Mansoor and Dr. Khalid Mahmood for organizing this extremely important dialogue. You know, as you mentioned, that this project is certainly a very important one, first of its kind in Pakistan, and that you have been successfully able to convince the policymakers to invest in this project. So certainly such consultation is extremely important because we would like to deliver. We would like to deliver on the promises, and we would like to deliver that science we intend to conduct would eventually help Pakistan in solving some of the critical problems in the field of nutrition and livestock. So it's something very important. I speak on behalf of comstech, which is the second largest bloc of countries in the world after United Nations, as a part of the OIC organization, and can tell you that similar problems are faced in very large number of low developing countries in OIC. I mean, out of 57 member states, about half of them have serious issues about malnutrition, and according to OIC, majority of the malnutrition, chronically malnutrition population, unfortunately, is in West Africa and in Sub-Saharan Africa, which lies under the OIC region. So I'm sure that this seminar is certainly not only of interest of Pakistan, but it has general importance. We understand that technology has played an extremely important role in preventing any major famine in last 5 decades. But situation seems to be very different today, as we have seen that technologies which help humanity to feed people have exhausted their potential, and human population is increasing with time. We have to feed very large number of people, up to the magnitude of about 9 billion in next 30 years, and certainly the available technologies or technologies in practice would not really be sufficient. So certainly this is an extremely important topic, and I'm absolutely delighted to see some of the leading experts in the field. We certainly looking forward to have their expertise benefiting not only Pakistan, but also OIC member states. And the proceeding of this seminar or workshop would help us to design a project which Dr. Shahid Mansoor has mentioned, something which is tangible and which can deliver on the promise. So with this, I would like to assure you that on behalf of Comstech and on behalf of the International Center for Chemical and Biological Sciences, where we have the largest genomic research facility, we would encourage all the collaboration within the country, bilateral and multilateral, and assure you that the outcome of this consultation would eventually lead to a very good project in which we promise to be an active partner. So thank you very much, Professor Khalid and Professor Shahid Mansoor, for putting together an excellent program. We're delighted to see our newest fellow of Royal Society, Professor Dr. Zulfikar Bhutta, speaking on topic which is very close to his heart, and he has been the leading expert in the field of human nutrition. So thank you very much, and I wish you all best of the proceeding of this seminar. Thank you.
Thank you so much, Professor Iqbal Chaudhry Saab, and with your huge support and leadership, I'm sure Pakistan is at a time now we need to take this the next jump, taking advantage of the technologies, and it's fantastic. So without further delay, I'll just move on to our next speaker. Our next speaker is Professor Dr. Mark Tester. Professor Mark Tester is working at KAUST University in Saudi Arabia. He's originally from Australia, and he's a plant physiologist with his experience working on a number of years on salinity. Soil salinity is a big problem in Pakistan, and just looking at the new innovations and how we can use this gene editing technology to address this challenge. So over to you, Professor Mark Tester. Thank you so much again for your time today for this event. Thank you. Over to you, sir.
Thank you very much, and thank you for the invitation to contribute to the start of this program. I have shared my screen. I hope you can see this adequately. Yes?
Yes, we can see it, yes.
Good, okay then. So I'm going to talk about the need for new innovative technologies for food security. I think this is clearly very important. We have new breeding technologies. There are a whole range of technologies, and I'll introduce other ones related in a minute. But I understand I need to focus here for this brief presentation on why we need them, to what do we need to direct our energies and our skills in new technologies. And for me, I'm going to give you the answer straight away, at least from my judgment, and previous speakers have already mentioned these: sustainability and human nutrition. Why not just yield? I think analyses are increasingly suggesting that we have, in fact, sufficient yield for a lot of the world in a lot of situations. In particular, we are eating, as humans on average globally, too much starch and too much red meat. And globally, when we analyze the requirements for increases or decreases in food production for 2050, if we were going to eat more healthily and waste less, there is, in fact, according to these analyses from the Eat Lancet Commission, no need to increase cereal production, or minimal need to increase cereal production between now and 2050. This isn't a generally popular, if you would like, analysis, but this is an interesting conclusion, even if quantitatively arguable. What is very clear from these analyses is that a lot of our energies would be better directed to increasing production of fruit and vegetables and legumes and nuts, so as well as fish, and decrease the emphasis on red meat production. And in fact, this decrease could even be greater if we are increasingly able to obtain protein from, for example, fermentation technologies. So focusing back on human nutrition to start with, it's very clear that our food system has a very big impact on our health through nutrient deficiencies, obesity, and even the onset and treatment of diseases. In particular, of course, biofortification has a massive role for improving human well-being. In our food systems, we have two major challenges. There's now more people going to bed obese than hungry, and the costs of obesity are spiraling very, very steeply in a lot of the world's population. Of course, of more major immediate relevance to Pakistan is malnutrition-related issues, where very large numbers of women and children are suffering from micronutrient deficiency. And we will hear a presentation later this afternoon from Yannicka Balk from the United Kingdom, doing some excellent work. There are technologies, and it would be wonderful if those could be deployed. Sustainability is another primary issue. Yes, we have a large area of land being used for agriculture, and it is no longer increasing and has not been increasing for quite a long time. So we do need to increase the outputs on what land we do have left. That is without doubt very important. But also very important is the fact that by far and away, most of the water that we use as humans is used for agriculture, and every aquifer that is being used for agricultural irrigation is depleting. This is particularly critical in Pakistan and India, where we have very large decreases in groundwater reserves. The irrigation systems that we're using at the moment are not sustainable. We really do have to be addressing this problem very, very seriously. So in Southeast Asia, in South Asia more broadly, even more than 70% of the water used is used for agriculture. So I think this irrigation systems need to be studied very, very carefully. It is particularly pertinent in Pakistan, but also, of course, in other countries in the immediate region. So some of these issues are pertinent for our neighbours as well as Pakistan directly. And this is becoming even sharper in the context of global change. And there are two articles in this month's Nature Climate Change that attracted my attention. One predicting that although water runoff from the Tibetan Plateau will increase, it is predicted that most of that increased runoff will be going into China. And in fact, in the Indus-Ganges basins, there will be a decrease in water runoff. So a problem which is already critical, I think, is going to become much more so in the next 10 to 20 years. Final issue of unsustainability of our agricultural systems are related, of course, to our CO2 emissions. Most of this is from methane. A lot of attention is given to the methane emissions from cattle, but equal to the, it is estimated that equal to the methane emissions from cattle are methane emissions from rice, wetland rice paddies. And I think research to improve, for example, dryland rice productivity could lead to really substantial contributions to reducing global CO2 equivalent emissions. I've already mentioned waste. The supply chains we are facing here in globally are clearly unsatisfactory. There is way too much food that is lost and wasted. These are quantitatively highly significant values. So we have major issues, but I'm an optimist, and I do see that we have many opportunities to address these because of technological innovations. Now, I'm just going to stretch a little bit beyond my remit to talk not just about biotech innovations, but also to remind us all to keep an eye on the immense power of digital technologies and innovations that they can provide for the food system. And although I've divided these into digital and biotech, they really do feed off each other and support each other. So, for example, in digital technologies, we have blockchain, machine learning, computer vision, sensors, IoT, robotics. These are all fabulous digital innovations, which really can also contribute to biotech innovations and the deployment of biotech innovations. But they're also powerful in their own right to reduce waste and loss, for example, in our food systems. And that's important. It also helps empower consumers and help consumers make more informed decisions when they're purchasing products. Not to mention, of course, just increasing production efficiency in its own right. So digital technologies, I think, are something that we all really need to make, really do need to keep front and center of our consciousness, in parallel with, of course, the biotech innovations, which are the primary focus of today's discussions. Genomics, genomic selection, very powerful for accelerating breeding. Phenotyping, using digital technologies often, can really help accelerate genetic gain in breeding systems. Genome editing has been mentioned, but we can also think about some of the other areas in the animal and meat protein production areas. GM, stem cells, continuous flow fermentation can be a technology now used for producing COVID vaccines, can be diverted afterwards to the production of protein using microbial systems. Neo-domestication, I think, is much more possible to generate and deliver step changes in our agricultural systems, such as, for example, developing seawater, saltwater-based agricultural systems for developing sustainable fish feedstocks. I've already mentioned non-animal protein, but of course, biotechnology can be very important for delivering fortified cereals. So I think one of the areas that are of great promise is the new genetics, where genomics is turbocharging genetics. And just as medicine is facing a revolution powered by genomics, so too is agriculture. And when we put that together with the powers of phenotyping and phenotyping in field environments, together we really have huge opportunities for serious innovation, both by accelerating the improvement of the crops that we currently have and unlocking wild germplasm, for example, through neo-domestication. So we can make existing crops, for example, more salt tolerant. We can also get salt tolerant plants and make them more like crops. Just one quick example as well. I'm very strongly in favor of enterprise, both for generating innovation and delivering innovation. And small businesses, I think, have a very important role. And one of our roles as decision makers and leaders is to help provide environments for allowing innovation delivery through enterprise. This is one example. Red Sea Farms, a company that I co-founded on the plant science side with an agricultural engineer to develop saltwater-based systems for the growing of crops. For example, to grow salt-tolerant tomatoes on diluted seawater in a greenhouse that's cooled with seawater and where the roof can be cleaned with no water, and developing a system where up to 95% of the fresh water that is normally used to produce tomatoes and other horticultural produce can be replaced with seawater. This is a drone image taken just a couple of weeks ago over some of our little field trial sites and our first pilot greenhouse, seawater greenhouse facility here on the KAUS campus. So we have technologies. We are needing to make decisions about how to direct our skills and energy. And I would commend to people to think very seriously about human nutrition and sustainability as two priority areas. Thank you very much.
Thank you so much, Professor Mark. It's very impressive and very thoughtful talk. And the thing that you mentioned that touched to our heart, especially taking the advantage of the, I would say, the smart technologies. And yes, you would be surprised to see Pakistan is the country which has the highest number of, I would say, smartphones penetration. So I think we need to use this technology to train our farmers, to inform our farmers. And you're absolutely right. We need to produce more per drop of water. And also, just to make another announcement, I forgot to mention in the beginning. So this event is live on YouTube as well. So there's a link in the chat box here. So if any of you wanted to share with your network, please do so, and also to students as well. So it's a great learning opportunity. So it's a great.
Khalid, yes, I mean, just because there are a lot of people who are unable to join, so please join. So by using YouTube channel. So
that's great.
Don't miss the seminar. Especially a lot of young students are calling me. So please join on YouTube. Thank you very much.
Thank you so much, Dr. Shahid. And also this event would be, it's recorded, it would be available afterward as well. So just to elaborate, because food is very important, and food is not coming from only wheat, not from carbohydrates, so looking at protein, looking at sort of aquaculture, and sort of all these other, I would say, the food web. I use this terminology, food web. So the soil is very important as well. So starting from the soil to the water, to the plant, to the livestock, to the humans, the ant as a food that come on our plate. So what's in there? I think that's what we need to learn now. And without wasting further time, I would like to invite our next speaker, Professor Zulfikar Ahmed Bhutta Sahab. He's the director and the founding member of setting up this institute in Aga Khan University, which is its first of its kind. It's Global Health and Development Center for Excellence for Women and Child Health. He's also holding a chair at Robert Harding Robert Harding Institute. Robert Harding. It's based in Canada. I'll ask them to come and teach us or tell us where the opportunities are, especially in Pakistan, where we have got more than 40% of the children, they are malnourished. Our food is deficient in those trace elements, which are playing important critical role in our food system, in our health system. And without further delay, over to Dr. Zulfikar Bhutta Sahab. Thank you so much for joining. Thank you.
Thank you very much, Dr. Mohammed and Dr. Shahid Mansoor, for this kind invitation. I hope you can hear me and you can see my slides on the screen.
Yes, we can hear you, sir.
Sure.
And can you see the slide?
Yes, we can see the slides as well.
Yes.
Wonderful. All right. So my task is straightforward, is to follow on the first opening speaker and give you the context of why your work is so important for Pakistan. I have for long admired the progress that the agriculture sector has made in Pakistan in science and technology, in newer methodologies, even in biofortification. But its translation into public health and its impact on nutrition is where we need to now move and work towards. So what I would like to do in the time allocated to me is to cover some of the information on what I'll share with you around the most recent information that my group was able to obtain from the National Nutrition Survey that we undertook recently, to give you 5 key messages from this survey that I think impact your work and that of other public health professionals, and then leave you with 5 key actions that I think we might be able to do to address this massive issue. So let me start by saying that the National Nutrition Survey that we undertook in 2018 and the beginning of 2019 was the largest ever such survey done anywhere in the world, on a survey looking at not only anthropometry and broad nutritional status, but also on micronutrients and on biomarkers. And this survey was also notable because it also integrated within it not only a poverty assessment, a food security assessment at household level, it also brought in dimensions of the environment. And it was a survey which also included elements of food, of water security and water safety. So over 100,000 households and an overall sample of children, which is close to around 67,000, representing various provinces of Pakistan. The survey was actually powered for district level specificity, and that's why the sample size is quite large. And we at the Aga Khan University and my own nutritional research lab were able to do it largely because of our laboratory network. Otherwise, maintaining a cold chain for some of these very sensitive micronutrient samples across a very diverse and challenging geography would have been very difficult. So we were able to do this and maintain a steady flow of samples to the university for assessment. We were able to undertake also on the spot bedside hemoglobin assessments using HemoQs. And as I mentioned, also samples of water, looking at water security and fortification status of some of the foods available at home, notably iodized salt. So what were the key findings from the survey? So the first unfortunate finding is that malnutrition rates, when you look at children under 5, haven't changed much in almost half a century. We are less than a century old, but in the entire period where we have been looking at malnutrition status of what used to be West Pakistan prior to 1971 and now is Pakistan as such, you will note that for stunting, over the last several decades, our rates have been stuck at around 40, 41%. And notably also wasting rates haven't changed very much overall. Some provinces may have progressed, but overall we are still stuck with fairly high figures. And this is not ubiquitous. It's not the same problem in every province of the same magnitude. When you look at the geospatial distribution of stunting levels across the country, it's largely a problem in the south, of the country. You will notice even in Punjab, southern Punjab has stunting rates in many of its districts as high as 40-45%. It is the problem in the south. If you look at provincial trends, you will very clearly see that over the last 9 years, since the last survey was done in 2011, there has been some progress in a few provinces, but by and large, many of the larger provinces like Sindh and Punjab have seen no change in stunting levels. Wasting, on the other hand, is a different issue. And very alarmingly, in some provinces, notably in Sindh towards the south, wasting rates have gone up. And wasting is a much more acute manifestation of undernutrition than stunting. So if you look at the distribution of wasting, no big surprise, it's also clustered towards the south of the country, notably in provinces of Balochistan, parts of Sindh, southern Punjab. But the most egregious, the most problematic, and perhaps from long-term outcomes, difficult form of stunting in children in Pakistan is where you have both combined wasting and stunting. So if you look at this concurrent wasting and stunting, it also clusters in these geographies. And this is important not only because it significantly compounds the risk of mortality. Children who are both stunted and wasted have about a 12-fold higher risk of mortality compared to those who are stunted alone or wasted alone. And it also has long-term developmental outcomes, which are adverse. The next key finding of the survey, something that we suspected, was that there is a very close correlation between maternal nutrition and childhood undernutrition. And in the interest of time, I'm only going to give you one or two snippets here. So if you look at which subgroup of mothers have highest rates of stunting in their offspring, in children, you will note that the blue band are those who are wasted themselves. So wasted mothers, those who are underweight with a body mass index under 18.5, have significantly higher rates of not only delivering babies who are small at birth, low birth weight, less than 5 pounds, but also are more stunted and also more underweight. And you see this clearly manifested in the overlap when you look at the distribution of maternal wasting or maternal low BMI and childhood rates of stunting and wasting. So it doesn't take a rocket scientist to work out. These are the same districts in Pakistan where you have very high rates of maternal undernutrition, that you also have concurrent child undernutrition reflected by stunting and wasting. So, I mean, these solutions are clear. If you need to do something about this here, you have to start with the mother. The third is that even if you had a good start, we have certain practices in our population which have nothing to do with poverty, but they have to do with practices. And they can be improved with significantly modest investments. So infant and young child nutrition, which largely reflects nutrition of young infants under 12 months of age or 24 months of age, if you take complementary feeding period into account, is something that I'd like to underscore as a big issue in Pakistan. Let's look at exclusive breastfeeding rates. Now, these rates overall may be slightly better than what the Demographic and Health Survey had shown, but still show a drop in exclusive breastfeeding in the first 6 months of life, which is the global norm and standard, from close to around 70% to less than 30% by the time they reach 6 months of age. And this depreciation reflects not that these children are eating solid foods, but they are being given all kinds of other products, like including teas, non-nutritive fluids, and animal milk. And sadly, in many provinces, what we found in the survey was that close to around a fifth of all children are never breastfed at all. The red band here in this graph shows you those who were never breastfed. And this is a hugely shocking thing. 1 out of 5 children don't receive their basic fundamental birthright of access to maternal milk. And this is something which is holding back not only the nutritional progress that the country is making, but also child survival, because there's a very close link between breastfeeding and child survival. When you go beyond the first 6 months and when children need to start complementary foods, which are dependent upon many staples and products that you're going to talk about, we also find that there is very little adequacy in terms of the diets that these children get. So both in choices and in diversity, this is a score called minimal dietary diversity that we use. And without going into the details, you can see that even in the relatively well-off provinces of Pakistan, like Punjab and parts of KP, there is very little dietary diversity. So that's very important for us to address from a policy perspective. The fourth is that you obviously need to have strategies. And in Pakistan, both for health and nutrition, primary care programs matter for nutrition, but they need to have good quality and good coverage. And I'll just illustrate that with one example. So if you take antenatal care, which mothers receive in pregnancy, and this is where they receive counseling, they receive immunization like tetanus toxoid, and they also importantly receive nutrition commodities like iron folic acid. And you look at which is the subgroup, and this is obviously controlled for poverty, has the better nutrition outcomes. You see in this analysis that whether it's for low birth weight, being born small, stunted, wasted, in every instance, the group that is the best is those that have received 4 or more visits, the minimum old WHO standard. Currently now we are requiring 8 or more visits in pregnancy, which is very challenging. But let's say leave 8 aside, just take 4 visits in pregnancy, antenatal care. That's the group that does much better. And if you can just flip this around and say those who did not receive antenatal care at all, the ochre one here, you will find that the outcomes in those subgroups who received minimal or just 1 or 2 visits was much worse. You also see this reflected in iron folic acid intake. So I just analyzed very simply nutritional status in relation to whether or not they had received iron folic acid in pregnancy. We did not even yet calculate whether they had received adequate iron folic acid in pregnancy. And consistently, those who did not receive iron folic acid in pregnancy had higher rates of stunting in their offspring and higher rates of low birth weight. So there is primary care programs make a difference. And this is what we need to do in our rural populations and also in our urban populations, urban slum. And the final point that I'd like to underscore is that although I have spoken about undernutrition, wasting, stunting, maternal malnutrition like low body mass index, Pakistan has made a nutrition transition, and we now have a yin and yang of emerging overweight and obesity, as well as persistent undernutrition in various geographies. So we have 2 things happening concurrently. But it's not just a double burden of malnutrition, it's actually a triple burden of malnutrition, because one thing that we're going to factor in is the prevalence of micronutrient deficiencies. I will only show you some data in the interest of time. But let's look at what is the relationship between underweight, overweight, and micronutrient deficits. So when you look at women of reproductive age, women between 15 and 49 years of age, and you look at what has happened in relation to the distribution of underweight and overweight obesity, you see a bimodal pattern. So you see, yeah, we have close to around 14-15% overall women who are underweight, less than BMI of 18.5. But about a quarter are overweight. And if you take obesity with a BMI greater than 30, then you can clearly see that just as many are underweight as many are overweight. And in many instances, you find them as parallel populations in the same geographies. Now, many people say, well, if you're poor, you should not be overweight. Well, the reality epidemiologically is that many poor populations also have very poor diets. And this is also reflective of poverty of choice in terms of inappropriate dietary consumption in many of our populations. And this is a massive issue. And it's a nutrition transition that takes place fairly rapidly. So when you look at the under 5 children, overweight and obesity are quite uncommon because it really does not manifest itself that early unless you are, in a very specific subgroup and on the right side of the curve. So whether you take underweight, stunting, etc., obesity rates overall in children under 5 are very low, and overweight rates reflected in this particular graphic are also rare. But what happens when they reach adolescence? So we have taken a separate survey with a school-age population, which I will not share today. But if you look at adolescence, which is 10 years to 19, and you look at what happens in Pakistan, overweight in girls, which is what I'm showing here, is almost universal again. And you see this in the same very geographies where you're beginning to see high rates of undernutrition. And this nutrition transition translates also into high rates of obesity in some of these very populations. And by adulthood, when you look at women of reproductive age, this becomes almost ubiquitous. So now you look at obesity rates and its prevalence in some of the relatively well-off provinces where underweight and stunting rates and short stature have gone down. And you see quite a significant clustering around northern Punjab of emerging obesity. Now, this has happened over time, and it has happened across the board, across all socioeconomic gradients. So this slide shows you the 5 asset quintiles, which is wealth quintiles, poorest in the light blue and the richest in the darkest blue. And you can see overall that in all subgroups there has been a change. It may be relatively less in the richest, but it is across all asset quintiles that we have had a change in BMI with increase in obesity and overweight. And these are also associated with micronutrient deficits. So very quickly, there was some of my final data slides. If you look at anemia prevalence in women of reproductive age, you see quite a significant preponderance towards the south of the country. And you also see that notably with iron deficiency anemia, where you have biomarkers of iron status like ferritin also measured. And this is widespread. It has nothing to do with socioeconomic status. It has a lot to do with dietary choices. And remember that we are not a vegetarian population. So it's interesting to see that the prevalence of anemia in our poorest populations in urban and rural settings are broadly comparable. There are not massive differences. And the point here is not to say that the poorest have highest rates of anemia, is to say even the relatively rich have anemia in about 38% of their population. So as we take all of these data in their entirety and we look at what we can conclude from the National Nutrition Survey, it is very clear that maternal child undernutrition, malnutrition in Pakistan is multifactorial. And it has complex socioeconomic, cultural, behavioral determinants, as you can see from the data presented. While the populations living in extreme poverty and food insecurity towards the south of the country are at greatest risk, malnutrition in all of its forms cuts across socioeconomic strata. And it's a dynamic situation across the country. We have a triple burden of malnutrition right now, with coexisting undernutrition, emerging issues of obesity, and ubiquitous micronutrient deficiencies, which really impact our human capital. In order to convert these data and now a lot of granular information into actions, we really require the political will. But I sometimes wonder, because this particular government right now has the political will, but it's taken now 3 years, and even now we don't have the federal nutrition program in place. So it takes more than political will. I think it requires a lot of skills in terms of translating policy into actionable things that can take place at the level of provinces. So we need a national nutrition action plan, and which is not on paper, which is actually implemented. And if Kausar Malik, my friend, is listening, he would know that we've been trying to do this for over 2 decades now, and at all levels, federal as well as provincial levels. And to get a real national nutrition plan in place would require partnerships with people like yourselves. So what can we do? So first thing to remember is that to address undernutrition, you need to start outside of nutrition sectors. So there are fundamental social determinants that have to be addressed in Pakistan. What we used to call as nutrition sensitive interventions, now we call them indirect interventions outside of nutrition. So education, prevention of early marriage, taking case of population growth, improving our environment, water sanitation in particular, and social safety nets to target extremely poor populations, which in Pakistan's case is about 1/3 overall. We need to strengthen existing health and nutrition actions, improve the mother's nutrition. If you want to start with a baby, you will miss out on a lot of things that need to happen during fetal development and pregnancy. The most inexpensive and obvious intervention is to get our breastfeeding rates up. And that requires a massive social mobilization, because the government cannot force people to breastfeed. Complementary feeding improvement, this is where you come in, because we need solutions from the food and agriculture sector, from our industrial sector, to produce low-cost complementary foods that children can be given. rather than the absolutely abysmal diets that they receive at home. So this requires dietary diversification and also low-cost product availability. Fortification, I'm a very strong proponent of fortification programs. We did a large national review which led to almost a $200 million investment on national fortification, which was just stopped because people just could not get it into the policy framework. But we require this for wheat, we require this for our food commodities, which have been there on paper, but the quality of fortification is variable. And we also need to do something about ubiquitous vitamin D deficiency. Children who are severely malnourished, some of that I've mentioned, do require treatment. So we need therapeutic strategies for those who are malnourished. And we need to consider bringing adolescents into this mix. I mean, young children, young girls in particular, who will be the mothers of tomorrow. We have the interventions. I've just published the whole Lancet series update on nutrition last week. And these interventions have been updated sequentially over the last 20 years, and they are available for people to put into programs. But we need to not only improve the coverage and scale, we need to improve the quality of what we are doing. So the antenatal care example I gave you, we need to make sure that those contacts are of good quality and they take place in every contact point within the health system. And we need to increase the agriculture and public health nutrition crosstalk. I firmly believe that this is something that we need right now, is to have scientists like yourself, and frankly, your biotechnology advancements in agriculture are far ahead of anything that we have in public health nutrition. Even linking some of the research that needs to take place around climate change, water security, soil quality, micronutrient deficiencies are ubiquitous. We know that our soil is depleted in zinc, yet our solutions in terms of biofortified crops with high zinc content, they're at our doorstep. That research has only just been done, but it hasn't been translated into large-scale policy or implementation. And of course, addressing food security through this. If you remember the maps that I showed you, some of the highest prevalence rates of stunting and wasting are in children living in the most agriculturally productive lands. And that has nothing to do with just pure food production or crop yield. It has to do with distribution pattern. So there has got to be also that link with access and also with poverty alleviation. This needs a national movement. It's not something that you and I can do alone. Behavior change, a national program for nutrition in which everybody participates. And as you heard from the preceding speaker, there are now global guidances available on what needs to be done, particularly for the double burden and etc. We have a poverty alleviation program that for the first time in decades now has got a conditionality around inclusion of nutrition. Within it, it's called Ehsaas Nashwa Numa program that Dr. Sania Nishtar is leading. And I think it has a lot of promise in terms of doing something for the poorest of the poor. And we need to address school-age children. So my current work right now, based on these surveys that we've done in Punjab and Sindh, is to try and see if we can do something for girls in particular in our schools, starting between 5 to 10 years of age, so that we don't miss out on a certain time period and just pick them in adolescence later on. So all of this, to finish, requires monitoring and evaluation. So it shouldn't be Zulfikar Bhutta coming out every 4 or 5 years and saying how bad things are. We need to have a process which is not dependent upon these periodic surveys. We need to be monitoring nutrition progress, just as we do for agriculture. Agriculture does not have periodic surveys. We have data on what is happening on a regular basis. We need the same for human nutrition in Pakistan, a nutrition dashboard that's populated by information from our district health information system that does not give you birth weight data on approximations, but on reality. And we need to produce this report every year. We need an annual national nutrition report, and taking advantage of Iqbal Chaudhry and Shahid Mansoor here, I would offer and suggest that the health and agriculture sector should produce a joint report every year on what is happening in terms of both our food availability, quality, and human nutrition. And we can do it if we put our heads together in terms of data sources and where that goes. So thank you very much for your attention and for giving me the time to speak to you today. And I wish you all the best in your deliberations.
So thanks a lot, Professor Zulfikar Bhutta. And you provide a very detailed insight on the status of human nutrition in Pakistan. And actually your guidelines could be very helpful for us to design the plant nutrition strategies, which is the main theme of this workshop. So I am Avais Rashid. I will be taking over as a moderator. So our next speaker in line is Dr. Lee Hickey. So Dr. Lee Hickey is Associate Professor at University of Queensland, Australia, and he has a strong interest in integration of breeding technologies, such as the rapid generation advancement technique, which is called speed breeding. And his advice for the speed breeding is sought internationally, and the technology is now adopted by the plant breeding programs all over the world. So today he will talk about the speed breeding and adoption of speed breeding by the several leading crop breeding programs all over the world. So Shamshad, can you please let the video in?
Hi, everyone. Thanks for this opportunity to give a short talk. I think the initiative in Pakistan to build speed breeding facilities is fantastic. It's a great pleasure to give a bit of a summary of the technology and how it's going around the world, how it's being adopted in different programs to accelerate plant breeding. The idea of speed breeding, well, I guess originally came from NASA-funded research that was done back in the 80s. And the way that they were growing wheat very fast was exposing the plants to continuous light. So the plants didn't sleep. It triggers early flowering, and so they could grow a very fast crop of wheat in space. The time lapse you're seeing is the footage of the first wheat crop grown on the International Space Station. And so my colleagues at UQ thought this was a great idea, potentially very useful for fast tracking our plant breeding efforts here on planet Earth. And so first started applying this to wheat breeding here at the University of Queensland back in 2003. So you can see it's certainly not a new technology. It's been around for a long time. However, methodology and protocols to enable scaling up this technology to a level that's really needed for plant breeding programs and big research programs was lacking. We were really inundated with a lot of requests from people around the world on how to actually go about doing this. And so this really got us thinking that there was a real need to publish the protocols for speed breeding. So we teamed up with researchers from the John Innes Centre and the University of Sydney to make these protocols available for the first time in 2018, really demonstrating this technology could be broadly applied across many crop species. In a nutshell, this is what's involved when it comes to speed breeding in wheat. Here's a summary of the protocol. So 22 hours light, just 2 hours of dark, temperatures of 22 degrees and 17 during the dark phase. And if we use this protocol, we can grow up to 6 generations of spring wheat per year compared to just 2 or 3 in a regular glasshouse. To show just how fast the plants grow, we captured a time lapse in collaboration with the Earlham Institute in the UK. You can see speed breeding plants here on the left versus normal conditions on the right. And the plants are growing very fast because we captured this essentially because seeing is believing. A lot of people didn't believe how fast the plants actually grow under these conditions. So you can see the plants have flowered now in speed breeding, starting to senesce and mature, and it's harvest time. And we don't have time to finish the video, unfortunately. Normal breeding is just too slow. And so this protocol that was originally developed for spring wheat translates and works very well for a whole bunch of long day and day neutral species. So crops like spring barley, Racopodium, the model, chickpea, canola, pea. And so the list is growing as different researchers start to test this protocol on their favorite crop. As a follow-up publication, late in 2018, we published a step-by-step guide for setting up small-scale and large-scale speed breeding facilities, really walking through the whole process. And even if your crop was not listed in the protocol, we did it in a way that it really guides you through this process, gets you thinking about the key things involved in terms of tailoring a protocol to your crop, and the things you need to think about as you start to go forward to set up a facility. So this is a great resource to look at if you're planning to build a facility. In this resource, it has all of the conditions in which and setup that we use here at the University of Queensland for speed breeding, but also at the John Innes Centre. And this really highlights the flexibility of the tool. Speed breeding comes in many forms and shapes. It really depends on the species that the speed breeding system is tailored for. And so it's a highly flexible tool. When we think about scaling up this technology, growing more plants in a small confined space is key to reducing costs per plant. And so we've got to think of this as really creating plant factories. And so even this example is for barley, grown at different densities, but even when barley is grown at a rate of 1,000 plants per square meter, they still produce enough seeds for the purpose of single seed descent in breeding programs or pre-breeding programs. So this really drives down the cost of the plant to just a few cents. If we weigh up the different options for developing inbred lines using different strategies, once we make crosses, here's the traditional path. Takes up to 7 years to develop F6 lines if we're growing 1 generation per year. Shuttle breeding, very famous strategy, first adopted by Dr. Norman Borlaug, adopted now widely across different crops around the world, achieves 2 generations a year by shuttling material to location, and has a big impact on the rate at which inbreds are developed, half the time, speed breeding even faster, and somewhat comparable to double haploids, but you have a number of benefits through speed breeding, including additional recombination. Speed breeding is highly flexible across germplasm, whereas double haploids, it sometimes can be quite germplasm specific. And in fact, some double haploid systems haven't been developed for some crops. And it can be quite laborious and expensive, double haploids, to implement at a large scale. If we look at where speed breeding fits into the breeder's equation, here we have the genetic gain over time equation. So we have these different elements that a plant breeder is constantly thinking about, tweaking and refining. These upper components, generally trying to increase them. Of course, the denominator of the equation, years per cycle, very powerful part of the equation to start to tweak. That's where speed breeding comes in, just like shuttle breeding to some degree. Speed breeding can really be tailored for any species. So it's just about a matter of optimizing the different growth conditions. So we're talking about parameters like day length, temperature, light quality, nutrient delivery. All of these things can be optimized for any species. For example, we're doing it at the moment for banana. So it really can be done. Here's just an example of our latest protocol for speed breeding short day species. Some results for sorghum and pearl millet using 10-hour days. So the plants receive 10 hours of light per day. We're able to rapidly cycle these tropical short day crops, 4 generations a year for sorghum and 5 for pearl millet. And we can scale this up at high density. Here's pearl millet growing at a density of 300 plants per square meter. So really, again, driving not only speed, but also efficiencies in terms of costs, which are really important. This paper was a nice example published recently by the group in the University of Hohenheim, which really showcases how plants respond to different light qualities. There's an opportunity there for closed or indoor speed breeding systems that can harness the power of light to manipulate plant growth. So some really nice results in this paper showing that plants respond differently to different wavelength compositions. And so we can manipulate plant height and flowering time even more. Some examples of different speed breeding facilities to give you an idea of how this sort of technology can be deployed at a big scale. And these slides have been provided by some commercial breeding operations. So this slide is from BASF, and this is a speed breeding warehouse. They do the speed breeding indoors. This is for wheat grown at 3 levels at high density. And so at the moment in their breeding program, they cycle wheat 2 or 3 generations a year in this platform. That meets the demand in terms of the breeding cycle that they're operating on. However, they can go faster if they want. So it's not just about doing 6 generations a year with this sort of technology. It's about fitting it in with your breeding scheme and gill testing programs. Another example is Corteva in India. This platform is for short day species, millet, pearl millet. And so you can see pretty low cost infrastructure, polyhouse design, fitted with blackout curtains to control photoperiod all year round, high density plantings, fully barcoded, fully integrated into their genotyping platforms. And so this is saving the company money as well, because they went from a very big team advancing populations outdoors to a small team that's efficient, it's fast, and this is the way commercial plant breeding is done. So we actually built a similar facility at Ikrasat in India. Here it is under construction as part of a Gates Foundation project. And so this included upgrading existing glasshouse facilities for speed breeding crops like chickpea, as well as short day species, sorghum and pearl millet. And inside this room, you'll see it can grow up to 50,000 pearl millet or sorghum plants at once. These are the blackout curtains drawn in the middle of the day for photoperiod control. So automation and streamlining these protocols are key. There's a lot of opportunities to integrate speed breeding and genomic selection. Of course, you get more bang for your buck. Here's a traditional breeding scheme. Here's what speed breeding can do. Can really shrink down this crossing and inbreeding phase to just 1 or 2 years for most species. However, here's what speed GS can do. If you integrate genomic selection, you can start the breeding cycle much sooner because you can make predictions for the next breeding cycle. And so we can start to think about how these technologies can be integrated in really innovative ways in which population improvement can proceed somewhat independent of the line development and variety release pipeline. So if you're interested, you can read more in our latest paper in the Current Opinion in Plant Biology, just published. So some take-home messages from my talk. Speed breeding systems can be developed for any species, and they have applications across discovery research, pre-breeding and breeding activities. And speed breeding platforms are pretty common in the private sector. It's about time that we have these sorts of facilities in our public breeding operations. And integrating these technologies is really key. So it's very easy to integrate speed breeding with marker-assisted selection for key genes. And there's real opportunities there to integrate this with genomic selection to make really big gains. I want to thank a small team of a small army of people from around the world who've contributed to our speed breeding protocols and our funding bodies here in Australia. Thank you very much.
So we are very grateful for Professor Lee Hickey for his participation in our webinar. So without any further delay, I will invite the next speaker in line. So Professor Jianlin Khan. So he is a leading livestock genomics specialist working at Chinese Academy of Agriculture Sciences, and he is also an adjunct scientist at International Livestock Research Institute, ILRI in China. So he has applied state-of-the-art genomics technologies in livestock breeding. So today he will be talking about the genomics and application of genomics tools for selection of superior livestock animals. So over to you, Professor Khan.
Yes, thank you. Thank you for the organizer. Although the notice is very short, but I have been working with Professor Head and also Dr. Awas for quite a few years. I'm happy just to share with you a very short presentation on what we do on the genomic selection for livestock. We can see from the end of last century, move to the 21st 20 of this century, we see really some sort of change in terms of how we could generate enough whole genome data for breeding. So people in the end of last century already see the, the whole genome sequence will change the landscape for livestock genomic research towards breeding. There are several papers, I just choosing a few Popular reviews start from 2007. The first by adding the latest one in 2019. Why I'm stop? Okay. So we can see there is a sort of what we call paradigm shifting animal breeding. We move in from end of last century to today. There's a shift from people, we have been working on in the later 1990s, try to understand what is sort of the quantitative trait, locus or nucleotides, try to characterize how the QTLs or the QTs are expressed, and also we have been trying to understand what's the role of the epigenetic effect. But moving into this century, there is already some practical work going on in terms of the application of gene or marker-assisted selection for using the GS model. Then the second layer would be the development of the genome selection models from the traditional blob, moving into the G-blob or the SNP blob, with the modern models that can generate the genomic estimate for breeding values. That's what we call the GEBVs today. And how we could use the genomic selection in livestock areas. They are basically because we know, in particular for the commercial livestock production, there are several well-defined breed. From like if we take the example of cattle, there's a dairy, there's a beef breed. Certainly, because for the dairy and the beef breed, there are clearly sort of the clear genetic background difference. So I think for the application of genomic selection, there's the issue of crossbreed genomic selection, and also the genome selection within a breed. I think this is something in particular for our discussion today, focusing on the issue in the developing country. We have to think more sort of the within breed genomic selection, because I would show you in the next slide what's the issue to apply these commercial available tools. So there are basically the two major opportunities for genomic selection in livestock. The first one would be say yes, the genomic selection can be easily combined with the modern reproductive technology that would reduce the generation length. For example, these, if we apply the multiple ovulation and embryo transfer technology, so that we could select the best embryo to produce the next generation. By doing so, we could really reduce the generation or to speed up, like what we hear from previous speaker, something similar what is in the plant genomic selection or plant breeding, what is also speeding breeding, we could call in livestock area by reducing the generation time. The second opportunity would be how we could improve the accuracy of the BV value through a sort of training protocol based on the commercial animals, because we know the commercial breed, they have been highly selected. The animals are more homogeneous among each other. Therefore, the genetic background are similar. The accuracy of these genomic-based breeding value are much more accurate compared to our indigenous populations. But we could for specific trait, for example, the meat quality or the growth rate or the milk yield. These are the STEM trait people we are trying to improve in either the commercial or the indigenous cows. So if we have an accurate the genome selection-based breeding value prediction models, we could somehow to transfer this knowledge and the models from commercial to our indigenous animals. This would be the way to one side to reduce the cost for phenotyping, for pedigree recording. This is really very much lack in our developing country system. How it work between what's the difference between the traditional selection in livestock compared to the what we call the genome selection. This is basically one slide to show you simply say for traditional selection, we also estimated the breeding value of individual cows for dairy trade. You can see, say yes, in our flock, the individuals perform very differently in terms of milk yield. But this, the traditional way of breeding value estimation based on, say, what's the inheritance, inheritability of the trait multiplied with the average value of across the herd by removing the individual productivity, so that we get the breeding value of individual animal, so that from there you can do your selection for the higher breeding value animals. This is basically you can see we need a lot of recording. Add the animals, this is basically we do not take into consideration of the genomic or the genetic data. We only look at the trait, the recording themselves. Therefore, this is only the phenotypic based estimation. They are far from what we think the accuracy. If we look at it, how the genomic selection may work, we will involving the genotyping and phenotyping. We try to combine the two set of data getting by all the different models in consideration of the background of your herd structure or your experiment designing. We could choose different models. From there, we could do the prediction of the genome selection-based breeding value of individual animals. This is basically the combination of the phenotypic data and also the genotyping data. If we look at this slide here, we by doing the genome selection in dairy cow, we could take into a reference population. This is the population of cows you can keep on station where you can have facility for good phenotyping. Let's say the milk yield record, the meat quality recording. Then we could do the genotyping of all the animals. While if we move into another dairy herd where this is probably our candidate population, another location where we cannot do an intensive phenotyping, but we could do genotyping by sampling a snapshot sampling one time of all the animals. Then by using the different static models, we could predict all these, the reference and our candidate populations, all the animals we put together, we estimate the all the individuals for the GEBV values. From there, we can ranking the individuals based on these the genomic selection breeding value. Then we could definitely split our animals. We could look at the source of animal. One side is from the reference population. We have the good phenotyping data. Otherwise, on the candidate population, we only have the genotyping data. We could look at it, how the animals are performed. If we could move some of the top performed animal based on the genotyping alone from the candidate population, can we move them into the breeding herd so that we could enrich the diversity of the breeding herd by this sort of the open scheme of the reference or the breeding herd to the candidate population. I think we could somehow try to control the issue of inbreeding, based on the genomic selection. So there are several challenges how we could apply the genomic selection in livestock breeding. You can see we need really a large training population so that we could achieve reasonably accurate the GEBV estimation. We definitely have to think the continuous accumulation of inbreeding in the breeding herd over generation. Say yes, because we are, for the reference population, we need a continuous phenotyping because the accuracy is based on your phenotyping quality. And also we see there, I think over the last 10, 20 years, we see in particularly the dairy side where the genomic selection has been, much more applied. We still say yes, even you using the same genetic marker, but these to come up with the breeding values measurement over different generation, the accuracy of the breeding value estimation can be reduced. We need to rebuild a new training population again. So there's also an issue of cost. I think for dairy side, the genomic selection, say for the bulls, are based on the daughters of the bulls, not the bulls themselves. So therefore, it really says there is a bias of this selection of the dairy bulls. If your accuracy are decreased, then there will be also the decrease in the genetic gain. We also already see these, although we have training population, we try to transfer, the estimate and the models to our candidate population. But to say yes, I think the current practice already shows the SNP estimate for one breed cannot be readily transferred to another breed. For example, the estimate coming from the Friesian cow, you cannot simply apply for selection for the estimation of the breeding value in the Jersey breed. So there's a call for breed-specific genomic selection scheme. This is, I think, we have to more a concern of our genetic background of our cows in different country, where I think in this case we talk about the Sahiwal, for example, in Pakistan. Clearly, Sahiwal has a different origin in terms of domestication and also a very different genetic background today, even compared to the close breeding. Let's say for Jersey, we cannot simply apply, these models, the genetic markers are discovered in other breed. We need to come up with our Sahiwal specific SNPs And also, we need to come up with the phenotyping data of Sahiwai, where from there we can go move into a sort of, say, the accurate estimation of breeding value using the both the phenotyping and genotyping data. So therefore, there's really an issue how we could apply this, all the knowledge and models developed based on the commercial breed into our indigenous breed in developing country. So as we see, say yes, if we think about, I think by closing my presentation, I would focus on more on the genotyping. Say yes, genotyping, in this case, we are talking the first choice of markers would be the SNP marker, the single nucleotide polymorphism, because this is a very stable inherited marker. They are distributed across the genome in high density. If some of them are located in the coding region, they may have to really say the functional consequences and phenotype trait. If they may be located nearby or within the QTL region, and the SNPs are suitable for high throughput genotyping. That's why the SNP are most favored, say, the genomic marker for genomic selection. So there are a lot of genotyping methods for SNP. I will not go into the detail, but by looking at the slide, you could see that the things like RFLP, the SSCP, and move down to the spectral metric-based SNP typing, or the high resolution melting SNP typing method. But this is basically the more the traditional method. They are low throughput. We are talking, say, you could genotyping the SNP marker of 10, 20, or close to 100 markers in one time using this technology. The high throughput genotyping of SNP will be involving the microarray or the SNP chip technology. The SNP chip technology based on how we could discover the genome-wide SNP, how we could select our trait associated SNPs, how we could validate the distribution of SNP in our targeted populations. We have to thinking the cost of the SNP chip. The more SNP you are included, the high cost is involved. So but if it is a low density across the genome, maybe the accuracy may be reduced. And if you want to develop your own SNP microarray, you have to think, say, you may have your specific SNPs, but you have to also include some common SNP which are built into already the commercial data set. This would, at the end, allow you to combine your SNP genotyping data with available data set, so that somewhere we could move into a sort of meta data analysis to improve our smaller data set. I think, say, yes, we have to be aware of this, the tournament bias of the SNPs. This is the issue, say, yes, the SNP you discovered in one population may not be much more informative in your candidate population. This is what the concern here. How you could choose your SNP array. This is definitely relevant to your specific target. If you're moving into a diversity study, or you want to carry out a genome-wide association study, or you want to go for genomic selection or pedigree analysis. So there are several issues you need to consider: the distribution, the density of these SNPs, the bias of the SNPs. There's say yes, how your specific SNP data set would may be able to combine with the existing data set or the other recommendation. I think the last is the cost effectiveness. This link to the density of the SNP genotyping cost, and also the performance of the these arrays. Currently, there are three most popular systems: Alpha Matrix, Illumina, and more this GeneSeq is taking over some of the Illumina technology already. So for each of these three systems, I think if you think about our livestock species, there are probably around 20, 30 different arrays from low density move up to high density coverage. So the cost is different. We have to take into consideration, see how we really can think of the application of the genomic selection in our developing country production system. I have been discussing this issue with Professor Shahid over the last 3-4 years, linking particular to the issue of Sahiwa breeding in Pakistan. The major problem we are facing is how to reduce the cost of genotyping. We are thinking, say, yes, this $1-2 tools for one sample, but so far seem to me the lower cost we may get for SNP array is around $10 to $15 per samples. But even for this, the company would request you to submit samples of around, say, 5,000 or 10,000 samples one time. They may give you this offer of $10 or $15. Otherwise, they may thinking still going back to $20, $30 per sample. So this is really the major issue we are facing. We need a sort of collaboration, how we could develop a low density SNP chip shared across, say, 3-5 breed in different countries, where we come up with a large number of samples for genotyping at same time. This is where we have to think. Otherwise, for a single breed, a single project, we cannot afford this $100,000 genotyping cost. This is the issue. I think by this sort of short my introduction on the genomic selection in livestock, I will close here. I'll stop. Thank you for your attention.
Thank you very much, Professor.
Thanks a lot, Professor Khan. So you kept your camera off during the talk. So many participants, they wanted to see you. So anyway, it was a very good talk on the genomic selection. And I think most of the points, they were also relevant to the genomic selection in plants, not only the livestock. So our next speaker is Dr. Janique Balk. She's leading an independent research group at John Innes Center. So her lab works on the uptake of iron and cellular developments, role of iron in cellular developments, and they use genetic and biochemical approaches to study the iron biosynthesis, iron transport, and iron storage. So today she will be talking on the cisgenetic studies to develop high iron wheat. So over to you, Professor Bal.
Thank you very much for this introduction. Can you see my screen?
Yes. Yes, we can see your screen.
Okay, excellent. Okay, so I'll, and actually I have to thank Professor Mark Testa as well, who I talked to last Friday, and he suggested I should present some of our recent data to your meeting. So we've been trying to develop cisgenic methods to increase the iron content in wheat. And I should very much acknowledge my collaborators. I couldn't have done it without them. So that's Cristobal Uwai, who is at the John Innes Center, Wendy Harwood, who does the transformation at the John Innes Center, and Alex Johnson, who did a PhD with Mark Tester, who is now at the University of Melbourne. So you're probably aware that iron and zinc are very unevenly distributed in cereal grains. So that's in rice, but also in wheat. So here is wheat as an example. And here we've cut open a grain and we stain for iron in blue. And you see that most of the iron is in the embryo and in the outer layer called the aileron. And here the endosperm, which is most of the grain, is white and contains very little iron. So with milling, and we see that later in the slides, a lot of the iron and also zinc, so zinc is in the same, enriched in the same bits, is removed. And here is another staining picture for the very high iron content in the aileron, but really nothing in the endosperm. So if we look at, because of that distribution, we see that wholemeal wheat has actually quite a good iron concentration of 30 microgram per gram. So for comparison, that's almost as high as in meat, but the white flour is very low in iron. If we then look what the range is in iron concentration in natural variation, so that could be used in principle for classical breeding, that goes from kind of 26 to almost 70 micrograms per gram iron. Talking to Harvest Plus, and I think most of you are aware of Harvest Plus as an organization who put a lot of effort and money and research into biofortification, this is the target that they want to see. So that's actually very close to the upper limit. So it will depend on the success of the breeding if that can be done. If you look at white flour, the natural variation is from kind of 5 to 16 microgram. And that is even the highest is too low to get a good iron nutrition from. So because of all the enormous progress in sequencing, so we've been taking advantage of that. And as you're aware, so a full genome sequence for wheat, there were already drafts earlier, but in 2018, we had really a full genome sequence of wheat. And so we used bioinformatics to find homologous genes in wheat that are involved in iron storage and transport. And this was based on what was already known in rice and Arabidopsis. Then, so we then ordered the genes. So nowadays you don't have to clone genes anymore, you just have them synthesized. And then we tested them in baker's yeast as a model organism to see, are they really involved in iron transport or storage? And because in baker's yeast is very fast, you can do that in about a month's time. And then we aim to change the expression pattern of these genes in wheat. And we try and do that with cisgenics. So we take a wheat gene in wheat, but some of it is still transgenics. But basically this is important, first of all, for us to get a proof of principle. And once we know it works, we can do gene editing as well. So James Connaughton, a postdoc in the lab, he started with the genes that encode vacuolar iron transporters. So they have a function in iron storage in the cell, for example, in the aileron, because that's where they're highly expressed in the aileron, but they're very low expressed in the endosperm. So we decided to put in front of the gene, so VIT is vacuolar iron transporter, and in front of it, we put a very strong promoter for endosperm expression. So this is the high molecular weight glutenin promoter. Then we used a nopalin synthase terminator, and we have a selection marker. So both the promoter and the TAVIT gene, they are from wheat, but other elements of this construct are not from wheat. So it's a kind of mixed cisgenic, transgenic construct. However, as I said, for us, it's very important to first see proof of principle. And very clearly, that was indeed, it became clear it worked. So this is a control grain I showed you before. This is the iron staining. This is one copy of the transgene. And you see here more blue is going into the endosperm And here, when we had a multi-copy line, we see a lot of iron all of a sudden in the endosperm. So then what we did is, because we're interested in the flour after all from wheat, so we milled the flour and we used roller milling rather than other forms of milling, because in the UK, this is what is the most common way of milling. And in roller milling, you separate out different qualities of white flour, and they're called break and reduction fractions. And then what you're left with is the whole meal. And so you see that in our Tavit wheat line, we get really good iron levels in these white flour fractions. On the other hand, we didn't see a change in zinc, maybe a tiny bit here, but not much. And here the line here is actually, so the UK, like Pakistan, is fortifying wheat flour with iron. In the UK, I believe it's a lot lower than Pakistan. Pakistan is probably around 30 milligram per kilo, but in the UK, it's 16.5 milligram per kilo. And you see that here in these white flour fractions, we're well above that fortification level. We also looked for if there was any changes in cadmium and lead, which are toxic, and we didn't see that. Although I have to say, we didn't grow the varieties yet on polluted soils. So since then, in 2019, we got permission to do a field trial. So this is here, and this is the wheat when it's very young. And then in August, it was harvested. Last year, we didn't do a field trial because of the whole COVID pandemic. But this year, in about 2 weeks or so, we'll be planting the next field trial. So what were the results? Well, this was the control wheat grown in the field. And this was the high iron line. And you see here the iron in blue. So it worked as well in the field outside. This is actually very alkaline soil. So plants usually have difficulty taking up iron from the soil, although wheat is very robust in this, and it worked really nicely. So we hope that this year the field trial gives the same result. So far we have only hand milled the flour. We haven't yet done the industrial roller milling, but we see in our lines a very clear increase compared to the control. So this is all very good, but as I showed you, there wasn't much increase in zinc. And the other thing that's very important for mineral uptake for humans is bioavailability. And here is a very nice several studies by Alex Johnson at the University of Melbourne, and one paper came out last year, is how you can use nicotinamide synthase to increase the bioavailability of both iron and zinc. This has already also been done in rice. So in rice, there are transgenic lines with increased iron, zinc, and increased bioavailability. And so in wheat, just overexpressing this gene doesn't have a big effect on iron, but it has a very good effect on bioavailability. So we took the construct we already had, and then we made also the one, we basically took the same gene cassette that Alex Johnson had used, and then we need a selection marker again. So the NAS gene, so first of all, we did look at the wheat genes, the wheat NAS genes, but there is quite a few of them, and we were not sure which one to choose. With Alex Johnson, together with Mark Tester, had showed that this one worked in wheat. So even though it's a rice gene, we decided to go for it. It's expressed everywhere, so it has the ubiquitin promoter, and so this is expressed in the roots, in the shoots, and in the grain, whereas Tavit is only expressed in the endosperm. And it's good to express it everywhere, because if there is more nicotinamide production, you mobilize iron and zinc and other metals around the whole plant, and it helps it to get into the grain. So these are the data we have from roller milled grain. This is from a very early line, but still I think it gives a good impression. So we have control wheat and we have the wheat overexpressing both. You see in the white flour fraction a really good increase in iron. So that is good. We see the effect of the FIT gene. But this time we also see a slight increase in zinc, which is very nice, or it's not slight. I mean, it's 25 micrograms per gram. That is pretty good. And we see more zinc in the Bren fractions. So we didn't see that before in just the fit line, but now we also see an increase in zinc. So this is where we are. Oh, I should mention we have measured the increase in nicotinamide. That's there. So this is very much in development. These are actually unpublished data that I wanted to share with you. Now, where next? So this is all a kind of cis transgenic strategy. And as you know, the regulation at the moment doesn't allow that in many countries. So we are also thinking about doing other things that could be tilling, so induced mutation. It could be gene editing. Now we know that these things are possible to get a lot more iron, to get a lot more zinc and more bioavailability. Can we do it with non-transgenic methods? And we've done an RNA-seq study of the TAVID grain, and one gene that jumped out to us was an iron-regulated transcription factor, IRO3. This transcription factor, for example, regulates a NAS gene. So what we want to do is knock out the IRO3, and that should automatically increase the expression of NAS, for example. And we know from the model organism Arabidopsis, when this is knocked out, you get more iron in the plant, although the seed was not being measured. So this is now what we're doing with gene editing and tilling mutations. So I want to leave it short just to introduce my research to you. So just to thank people in my lab. So we have people working on other aspects of iron sensing, but we have people who work on the wheat project, especially James Connaughton. And then we work together with people from the Norwich Medical School for bioavailability. We work with people who image the iron. We work with people at Rothamsted, like Peter Shuri. And as I already mentioned, Cristobal Uwaya, the John Innes Center, and the wheat transformation platform. And I forgot to put here Alex Johnson. So thank you very much for your attention.
So thanks a lot, Dr. Janique. And I think it is very important aspect to improve the iron biofortification, because we have examples of the zinc biofortification in Pakistan, but I think iron biofortification is quite challenging compared to zinc biofortification. So thanks a lot. So our next speaker in line is Dr. Kamarpal Dhugga. So Dr. Dhugga worked most of his career in the industry for developing improved maize varieties. So now he is leading the biotechnology lab at the CIMMYT, Mexico, and he is involved in gene-edited maize and wheat varieties. So in our last workshop in Comstech in 2019, Dr. Dhugga presented some of his work about the gene editing in maize and wheat. So we are expecting now that he can provide us some more updates about his work on gene editing of maize and wheat. So over to you, Dr. Kamarpal.
All right, thank you, Awas. Let me pull my slides. Thank you for giving me the opportunity to share our progress, future progress with the audience. Okay, you see my slides okay?
Yes. Yes, we can see.
Okay, so what Awas asked me to do was to talk on, unlike the previous talks, which were more global and kind of like more, let's say, policy-based type of presentations. Mine is almost purely scientific. So Awas wanted to me to discuss how what we do in CIMMYT could be applied or could be used in Pakistan. Actually, in fact, I have been in talks with him for a while. I need to keep track of time because we're running way behind and I need to finish on time. I'll catch up some of the time that we have lost anyway. So, we have been in discussions and we have been trying to get some lines from Pakistan to CIMMYT that we can collaborate. Just broadly speaking, I want to put that up front. But let's go through what I have to say. Last year, ICRAR Khan, my former classmate from University of California days, graduate school. He asked me to speak in a webinar, and I made the slide specifically for Pakistan with Avas's help. Actually, he was the one who sent me some of the data. So what you see on the left-hand side is the genetic gain of the last 10 or 12 years in Pakistan. And on the right is global genetic gain in wheat. Wheat is the life and blood of Pakistan, we can say, or life and bread of Pakistan. So you see that you can look at the detail if you want, but look at the bottom line, which is that the rate of gain in Pakistan has been about 2/3 of that of the global annual rate of gain. So it's kind of like right there's about 30 to 35% opportunity, more gain in the bin, so to say, just if the genetic gain could be kept up. And part of the reason this genetic gain is low is just because of varietal replacement. I mean, the farmers, traditionally, I myself grew up at a farm, so I know what it is like. We tend to hold on to the varieties that do well once, and then we hold on to them for sometimes for too long, for a whole lifetime, so to say. So what happens with older varieties is that they become more susceptible to diseases as time progresses because they don't have this genes for resistance to newer races of the diseases. So this is one area where I have focused the work of my lab, which is disease resistance, because it was both the trait has to be simple for us to have any impact from gene editing. So let me go to the next slide. Okay, so a couple of years ago, Brenda Wolf and I wrote a commentary on this GM wheat, but I modified the slide from that commentary on the, essentially you heard about speed breeding earlier. So gene editing reduces the time further, but with one caveat, that if we edit the gene directly in elite lines, that's where I want to emphasize we stand out. I am not using any experimental lines in my work, because then you go back to this upper part, which is you go back to traditional breeding. You have to introgress that gene from a experimental line into your elite line. So what's the point? The introgression, number one, it takes time, like traditional breeding, and then with speed breeding even, anywhere from 5 to 7 to 10 years. But with gene editing, if we do it in the elite line, within a year to year and a half, at most 2 years, we have a product ready, because all you need to do is get rid of the editing machinery, and your variety is ready. You have left the genome more or less intact. There's this background editing and all that, but that's almost a minor issue or a non-issue. So that is the main crux of my lab. Editing genes directly in elite lines, period. So no genetic background dilution or contamination of anything. Okay, so at CIMMYT, I decided to focus on 2 areas in maize and wheat, both disease resistance. And these are both real life problems, because I came up with these traits after prolonged discussions with our breeders. In maize, maize lethal necrosis, I don't know how many of you have heard of this trait. This is a viral disease caused primarily by, and I'll go to that in a minute. And that in wheat, rust resistance, mainly primarily LR67, which is the hexose transporter, inactive hexose transporter, so makes it easier to address by gene editing, because all we need to knock out. And powdery mildew is beginning to show up both in northern Pakistan and in northern India. So that is controlled by 3 copies of this gene in all three genomes, and each one of them has to be knocked out. It's a dominant repressor of resistance. So both these traits are addressable by SDN1 scenario of gene editing. SDN1 stands for side-directed nuclear scenario 1, which is essentially a non-homologous end joining component of gene editing, and that causes errors in the frame shift and the gene. Essentially, it's a way to knock out genes. Okay, so let me give you some background. So MLN, maize lethal necrosis, is caused primarily by this maize clotted mortal virus in combination in general with one of the partyviruses. It showed up in Kenya about 2011 and in China as well, actually. It originated in Peru way back in the 70s, and then it was quarantined and all that. But once it showed up in Kenya, it essentially devastated the crop, maize crop. And maize is a staple in Eastern Africa. So most of Africa, let's say, but in Eastern Africa, where this is where this disease showed up. So in general, the reduction in yield or production, let's say, not yield, average reduction in production in Kenya was about 25%. And you can imagine taking 1/4 of the produce out of circulation. So it's a major disease, very significant disease, and we requested Gates Foundation to give us some money. They gave us some money, and we are working on that now. So I'm gonna make okay, here's an example. So the same hybrid, it's a heat tolerant and drought tolerant hybrid, grown in a normal environment, disease-free. Kiboko is on the other side of, the opposite direction of Naivasha. Naivasha is where our nursery is in Kenya. These are both in Kenya, where our maize lethal necrosis nursery, because it has to be a contained nursery, it has to be quarantined as well. So nothing comes out of that nursery. So here on the left, you see a picture of how the different genotypes look like. And we artificially inoculate the plants and screen them. Same hybrid. I don't think I need to say much under normal conditions and in the presence of MLN. So we identified a source of resistance in an exotic line. This line came from Thailand, Castor University, 23, very strong source of resistance around on chromosome 6. It explains about 50 to 60% of the variation in disease resistance. So here you see an example. We find mapped that QTL to about, now we are down to 100 kV region. I'll show you, tell you a bit more about that. And in parallel with forward breeding, we introduce that QTL into, this is the same exact line, 186 CKDHL double haploline from Simit, Kenya. And after 3 back crosses, you see the difference in the absence of the allele and in the presence of the allele. Very dramatic, very strong allele. So we fine mapped it to 100 kV region. And now we are down to a point where further combination is difficult. So we have to just go in and see which of these genes or non-genes is responsible, is the causal gene. So all these double arrows and single arrows are frame shift mutations, SDN1, and double arrows connected with a line, they are dropouts, which is that you have 2 guides to drop out the gene. So we have overall 15 different, what we call projects, which is this whole region has been divided into 15 different parts based on various reasoning. And we have obtained every single, the edited allele of every single one of them that we are currently screening in the greenhouse. So it's going on right now as we talk. And one thing I must say that the success, major reason is your partners. We have been collaborating with Corteva AgriScience, my former employer. I spent 2 decades at Corteva before I went to Simit, so that makes it easier to work with that organization. And this is a remarkable technical success. This is an elite wheat line. This is not, CML 536 is one of the parents of the crosses you saw, those hybrids earlier. And then in parallel, I think I'll skip this, just quickly give you a view that viruses don't have much of their own translation machinery. So what they do is once they attack the plant, they hijack the plant's translation machinery. And how they do it is by fooling some of the proteins that initiate protein formation, synthesis or translation from the mRNA. And eukaryotic messages, they have this cap on the 5' end. That is a signal for this machinery to initiate protein formation. Viral RNAs don't have a cap. So what they do is they fool this particular protein, 4E. It's an initiation, it's called eukaryotic initiation, protein initiation factor, or EIF. Eukaryotic EIF, eukaryotic initiation factor for protein formation. So virus particularly fools this protein, binds this particular protein, and the cap, it kind of like overcomes this lack of cap and makes the plant machinery translates its own RNA. So what has happened in the plants is that this protein has been mutated or knocked out. And there are other copies of the protein that the virus does not recognize. So that confers virus resistance. So we have employed that particular method as well in parallel. And there are 4 genes in the maize that very closely match with the 2 that are known to confer virus resistance in barley and other species. So we have knocked every one of these genes out. And that happened just within 1 year. That tells you about the technology. I want to keep emphasizing this is having good partners, great partners, because we as a CIMMYT, a small organization, we just don't have the wherewithal to develop this kind of technology. So Corteva, I mean, that's their living, that's their, I mean, that's essentially what they do as a big corporation. They have a close to $1 billion in R&D budget, and you can compare that to any other organization for that matter. So their technology, their tools are the ones that are helping us. 100% editing efficiency. Every single event we generated. And this means that if we started with 100 embryos, we generated 100 events, and all 100 were edited. Some homozygous, some heterozygous, some were somatic, but they were all edited. So we have plenty of choice. All we need is 1 or 2 events, and we have more than we need to move forward. So every single one is edited. So we are right about here because we started this whole thing last year in the beginning of 2020. And right now we are testing these lines as well and these are in 2 commercial lines, 546, 536, and 543. So these lines are being tested in the greenhouse now for disease resistance. Okay, how about wheat? This is my last slide, I think. LR67, LEAFR 67. That is a hexose transporter that came out of Graham Moore and Laguda's laboratories a few years ago. It's a knockout again. It's an inactivated form of a hexose transporter that confers resistance against a broad spectrum of fungal organisms. And that too is amenable to SDN1 scenario, or you see knockout by gene editing. And we are addressing this in an elite line, Reedling, which is Borlog 100. And what Avas was gonna do was gonna send me some lines that lack both LR34 and LR46, which are 2 of the most commonly deployed genes for rust resistance, so that we could test LR67, which is not present in any of the elite lines. This is present in ancient lines. And Avas told me actually that it originated in Pakistan. So in ancient lines. So what we want to do is use those relatively elite lines that Avas has that lack both LF34 and LF46 so that there is no background resistance. So we can, when we knock out LF67 gene, we can determine whether how much resistance is conferred. So we are still in the process of transferring those lines to CIMMYT. And this is our future. So I think with that, this is just the timeline, how much time it saved. This is how, I mean, the gene editing, I showed that in the second slide or so, that this is actually from the horse's mouth directly. This slide is from Corteva of actual product that they released, waxy corn. And you can see that with traditional breeding, and that's company breeding, it's about, traditional breeding is 8 to 9 years, 9 to 10 years. With transgene, you have to seek all those approvals, it's 13 to 20 years. And with gene editing, they actually did it. They had the product ready in 42 months. So with that, I rest my case. I think I have already said how we can collaborate with Pakistan. And I look forward to working with you guys. And also one thing I want to point out, I think I'll probably save it for another talk, which is on nutrition. You heard a beautiful presentation on plant nutrition. We have quite a bit of activity on the phytic acid side, because when we talk of nutrition, we talk of zinc and iron, divalent cations. And phytic acid is the chelator. It chelates both zinc, iron, and almost any other metal ion for that matter. And the available zinc and iron actually is not the same as the actual concentration of zinc and iron. So the ratio of iron, zinc and iron to phytic acid or vice versa is what determines it. So we have quite a bit of activity both on high throughput measurement of phytic acid for direct selection, as well as gene editing to reduce the amount of phytic acid. And from natural variation, we know that we can reduce it by 50% without any impact on seed health. With that, I rest my case, and I will take any questions when this whole thing is done. I think I was pretty much on time. Okay, thank you.
So thanks a lot, Dr. Kanwarpal. And we are sorry for any inconvenience because you have to wake up very early to participate.
That's okay.
So we look forward to collaborate more effectively with you. So thanks a lot.
Sure thing, yeah.
So,
thank you.
Next is my own presentation. So let me share.
Okay, Wes, I can skip mine, or do you want me to present something? Okay, so mine is at the end, sorry.
Okay. Yes, yours, next in line. Okay. So can you see the slides?
Yes.
Okay. So I will be talking about the new genotyping platforms for improved selection in crop breeding. So actually this presentation is more focused on wheat breeding, but these genotyping platforms which we have developed over the time, they can be taken as an example for the other crops. So Dr. Duga very well presented the rate of genetic gain for wheat in Pakistan, and that is the basic equation to calculate the rate of genetic gain. And any increase in the rate of genetic gain is possible if we make efforts in all of its components. For example, we have to improve the selection intensity. We have to select more number of individuals with our desired phenotypes. We have to improve our selection accuracy. We have to add more effective genes in our breeding programs through crop wild relatives or through land races or from other sources. And then we have to reduce the generation time. So we have presented this integrated approach to accelerate the rate of genetic gain. So it is basically an integrated approach. We have to move towards the more effective genotyping technologies. We have to adapt rapid generation advancement techniques. We have to bring in a better breeding strategy, which could be the genomic selection or gene editing or the combination of both genomic selections and gene editing. And we need to improve the phenotyping methods. So by the combination, by the integration of all these technologies, we can accelerate the rate of genetic gain. So what we have been doing over the time, so first of all, we have to learn about the genotyping requirements in any crop breeding programs. So we have three major scenarios. So in first scenario, we have very large number of samples, which we have to genotype very few markers. So this is the case of gene introgressions, marker-assisted recurrent selection, or gene tagging. So for this type of scenario, we have the uniplex marker technologies, and like CASP and STARP, we have very high throughput platforms which are quite flexible. And in these flexible platforms, you can genotype any number of samples with any number of markers. So the second scenario is where we have to genotype many samples with many markers. So this is usually in the case of diversity studies, association mapping, QTL mapping, and in case of genomic selection. So in this case, we need multiplex, high throughput or non-flexible platforms. And we have such platforms in the like the SNP chips and genotyping by sequencing methods. Similarly, we have another case where we have to genotype very few samples with many markers. So this usually happens in the background selection, marker-assisted backcrossing, or the screening the advanced material. So even for this case, we also need multiplex and high throughput genotyping platforms. And the examples are genotyping by targeted sequencing, which we can do by the next generation sequencing. So key platforms which we have developed over the time with the help of Chinese Academy of Agriculture Sciences and the CIMMYT, that we have developed very high throughut CASP markers, which have ability to screen the breeding populations for very important functional genes. We have developed a 55K SNP array, which is a new SNP chip, and the performance or the biasness of this chip is very improved compared to existing chips, which are like most extensively used SNP chip is the 90K SNP chip. At the third level, we have developed a genotyping by targeted sequencing platform, which can targetedly sequence almost 202 SNP sites. And I think this platform has very good utility in the wheat breeding. So similarly, to reduce the cost of SNP chip, we have developed another targeted sequencing platform which has 40,000 SNPs. And this can be used as a replacement of SNP chips, and the cost is almost 30 to 40% lower compared to SNP arrays. And then we are developing some genotyping or sequencing platforms for the gene discovery studies, and I will give you a detail in the later slide. So this is the framework where we developed the high throughput CASP markers. So after development, we validated and we proved that these markers are very effective, and these markers represent the different phenotypic variations in the crop germplasm. So this is the detail that if we use the CASP markers compared to the conventional gel-based markers, the high throughput is very high. For example, in the conventional gel-based markers, the total data points in a year which we can get are 0.1 million. But in case of CASP markers, this throughput can be reached to 9.2 million to 36.8 million data points in a year. And the cost is quite competitive. So we developed these CASP markers in 2016, and after their development and publications, we get feedback from several researchers around the world. And at this time, almost 40 genetics and breeding programs all over the world, they are using our CASP markers in wheat genetics and breeding. So another aspect is how we can reduce, because CASP is a proprietary product. So its cost, the cost of the master mix is quite high. So then we developed a strategy where we can use any type of master mix for high throughput SNP genotyping. And this technology is called STARP. So we converted all the wheat CASP markers into the STARP markers. And if you use the SNP markers in STARP format, you can further reduce the cost by 2 to 3 times. So second important platform is the SNP chips. Previously, several SNP chips were available, But the main problem were that they were very low density chips when we apply those SNP markers in other genetic backgrounds. So here is an example that if we apply those SNP chips, for example, 90K SNP chip in Chinese mini-core collection, only 18,000 markers were polymorphic out of 81,000. So they have very uneven distribution. They were not aligned to recent reference wheat genome. They have very high ascertainment bias, and there was no information available if these SNPs are associated with important phenotypes. So based upon these problems, we developed our new 55K SNP chip, which we call as trait breed SNP chip. They have very high quality SNP markers from all the chips which are currently available. They are uniquely polymorphic to all the genetic backgrounds. They have almost equal marker density on all the chromosomes, and they also include SNPs from the functional genes. And the cost is quite competitive. It is around $30 per sample. And this SNP chip can be used for the background selection, for the genetic diversity, for the genomic selection and GWAS. So this is the marker coverage for our 55K SNP array. So it has 40 to 80% more number of SNPs on D genome, because there is a genetic bottleneck on the D genome, and usually we have very low polymorphic percentage in wheat D genome. So the third aspect is how we can design more targeted GBS platforms, because the problem in SNP markers or the Cas marker is that they cannot be used for SNP discovery. We can use those markers for SNP genotyping, but we cannot discover if there are more SNPs in the flanking region. So for this strategy, for this problem to overcome, we designed NGS-based platforms. So they target the flanking region where we want to genotype the SNP, and then we use NGS approaches to genotype or call the SNPs. So library preparation for these targeted GBS platforms is very cost effective, and they are excellent platforms to identify new SNPs in any crop or livestock species. So we call this platform as a Triticum gene sizer. It is a allele discovery platform. So for example, in the first step, we targeted the 250 base pair fragment around the SNP. We targeted almost 200 genes. In addition to these 200 functional genes, we also targeted 63 loci in the wheat genome, which were neutral. So we can also use this platform for the genetic diversity studies, because to estimate the genetic diversity, we need the genetic regions or the chromosomal regions which are little bit neutral compared to the functional genes. So we are sequencing these regions at the 500x depth, and the cost for this platform is quite low, around $10 per sample. And this is very excellent platform for the gene characterization, especially when you are using the wheat genetic resources. So after development of this GBTS, this is called genotyping by targeted sequencing. So in the first step, we developed this platform for the 200 SNPs. And as I told you in the previous slide that these 200 SNPs include the 150 SNPs from the functional genes and 50 SNPs from the neutral sites on the chromosomes. So this is very excellent platform for gene tagging, and the cost is around $10 per sample. Then we developed a targeted platform for the 40,000 SNPs. All these SNPs are aligned to reference feed genome, and this also includes the SNPs from the functional genes. So this 40K GBTS platform is very excellent for the diversity studies, GWAS, QTL mapping, and genomic selection. And I told you that superiority is that we can also discover some new SNPs. So now we are improving this first GBTS 200 platform, and now we are developing a GBTS 500 platform. So what we are doing, we have included the previous 150 SNPs from the functional genes, plus we have selected 250 QTLs for the important breeding traits in wheat, because there are a lot of QTL studies available where the researchers have used the SNP markers. So we have selected those QTLs, and we have included those QTL markers in GBTS 500 platform. Plus we have increased the number of neutral markers, neutral SNPs. So I think GBTS 500 will be a very good improvement in GBS 200, and the cost will remain the same. So the last one is the biofortification GBTS platform, because we also want to use these type of platforms, these type of products or innovations for the gene discovery. And now, because we have the reference wheat genome available, we can predict some of the genes which have some putative roles in our desired traits. For example, here what we did, that we have identified almost 100 genes in the wheat reference genome which have putative function for the biofortification. And for these 100 genes, we have developed a targeted GBS platform. So this platform is under development. And once it is developed, we can use this platform to identify the variations present in our populations. And then we can associate this with the biofortification data. And then we can pinpoint the genetic variations which are associated with biofortification. And then we can use this information in wheat breeding. So that is the last platform which is under development for the targeted GBS platform for biofortification. So this is a speed breeding facility currently under development at NARC Pakistan, and hopefully it is funded by the PSDP and National Agriculture Research Center. They are responsible for its management, and hopefully this facility will be available for the generation advancement very soon in 1 to 2 months. So in summary, the Cas markers, they are very good uniplex markers. They provide you flexibility and they don't compromise the high throughput, and they can be excellent choice for some of the objectives in wheat breeding. So 55K chip which we developed with the help of Chinese Academy of Agriculture Sciences, it is very good improvement to the existing S&P chips. The targeted GBS platforms which we have developed for several of the objectives, they are very good platforms for the genotype to phenotype associations, for gene tagging, and they can give a very good, they can be used as a very good tool for improvement of selection in wheat breeding. So all the future crop and livestock breeding programs need sophisticated and very efficient genotyping technologies. So thanks a lot. So now we will move towards the last speaker of our webinar. So I will ask Dr. Shahid Mansoor to give his talk on the need of new initiatives in Pakistan to accelerate the rate of genetic gain. So over to you, Dr. Shahid.
Yeah, I must thank all the audience, the site speakers, that they have been very patient and listening to. So we are sorry to some of our audience that it has taken a little bit longer than we expected, but this happens when you have multiple speakers. So what I've suggested to Avais is that probably we can have some more targeted webinars in future about genotyping platform and some other on genome editing, so that people can have more time to ask questions. And so I will take a few minutes just to tell that what progress we are making at NIPG. So we met 2 years ago, and then last year we were in Islamabad, and what we have done so far is that we have come up with the we basically because of the program that we initiated in 3 areas: genome editing. So we are doing genome editing in rice, potato, wheat, cotton, and canola, and then genomics for. And one of the very important message which was from Professor Hahn was that we need to sequence genomes of all our cattle and buffalo breed, which have been done in collaboration of Chinese friends, and then genomic selection in rice, and then speed breeding facilities that we are developing. So this is the first success story which we have already published, where we have developed genome-edited rice with resistance to petty light, and where what we have done is that we have targeted switch protein promoter. This is one of the key genes required by the pathogen. In the second example, we have edited a gene for this is basically a point mutation, which was by template directed CRISPR editing, and we have developed herbicide tolerance, and this herbicide tolerance is very important trait for when we want to grow rice with less water. In another example, we have developed potato virus Y resistance, and as Professor Duga was telling us that there are some elongation factors which are important for the to enter into the plant and cause disease.
Sorry, Dr. Shahid.
Yes.
Dr. Shahid,
Yes.
can you speak close to the mic?
Okay, so thank you. So I think that what we have done is that we have done genome editing in potato, and we recently got some data to show that these edited lines are resistant to PBY, and these plants are in the glasshouse. And then there's also some of the other things that we are doing is viral interference, and then because of the time, we'll not go into the detail. So I will skip some of those slides, and then we are working with virus resistance. Again, I will step. There's another. So a lot of people talked about, especially Professor Duga and Awais, and then Professor Bach from UK. So they talked about, so what we are doing is that, we are trying an integrated approach of involving breeding and CRISPR-mediated genome editing to improve wheat yield. So the wheat yield is a very important trait. So we have identified some of the negative regulators of yield, like, those which are regulating the size of the grain size, or number of grains, or number of productive tillers. So some of the genes have been targeted by genome editing. So we are waiting for the final outcome of this editing. Then we are also repurpose some of those genome editing tools for the detection of SARS-CoV-2 virus. And then something about speed breeding. We have developed our own little speed breeding facility. We have developed a state-of-the-art speed breeding facility, but we are waiting for the fund. But in the meantime, we converted one of the room, and we showed that within 2 months or 65 days, we could develop seed. This is another example of breeding, genomic selection, where we incorporated a bacterial blight resistance gene from non-basmati into basmati rice, and by using the genomic approach, we were able to recover the basmati genome. And then finally, we have sequenced all the genomes of the indigenous cattle breed, as I pointed out earlier. Professor Han talk, if you remember that, we need to have the genome sequences as well as the diversity of our local breed, because the available chips are not good for our Indian breed. So we have done this, and because of this extensive work, now we are part of the 1,000 Bull Genome Initiative. Similarly, we have sequenced all the buffalo, because in our region we love buffalo milk because of the thick and creamy taste. So we work very closely with the breeder association in this work, and one of the very simple genomic selection demand was to find out the beta casein variant. This was work which was required by some of our breeding breeders, and this was a simple test that we developed, and we are providing the service, and more than and many breeders from throughout the country are using this facility. And then finally, we are, as Professor Han has discussed, there are important constraint about the genomic selection in our breed. So what we are propose is that we can have a low density SNP chip of our own, where we can have breed specific marker for the breed which are found in Pakistan, as well as genomic selection. And I think that, because of the use of this our own chip, we hope that we can provide a cheaper service to our breeders in livestock. With this, I must thank all the audience, especially my colleagues who have on a very short notice, they have agreed to give their presentation, and I'm for the students and for young researcher, I must say that we will have a series of smaller, very targeted lectures, so that our younger colleagues can ask questions, and also they can have more interactive. We actually designed another workshop last year, but due to COVID situation, we were unable to host this. It was supposed to be in Turkey, but I hope that with the improvement of situation, we will be able to move to have more session in person. Thank you very much. Wes, do you want to continue?
Yeah, I think if someone has question, they can send by chat. So I could not find any specific question.
Yeah, then we can invite, we have Dr. Johar and Dr. Kausar Abdullah Malik for their comments. We have kept this whole session technical, and we have so Dr. Johar, can you unmute? Can we unmute Dr. Johar? Dr. Johar? Dr. Kausar Saab, if they are available? Can somebody unmute Dr. Kausar Abdullah Malik?
Yes, I have been unmuted now.
Okay. Thank you very much, sir. I think that probably you are the mentor of all these activities. So we take a lot of pride having you in all our activities. So thank you for being with us for a long session. So over to you, sir.
Thank you, Shahid. Thank you. You see, I have very attentively, listened to all the talks, you see, and I am delighted at the quality and the content of all these lectures and talks. As you know, that we belong to the era of agrobacterium, you see, and I'm so happy that the things have moved so fast, you see, and now we have possibilities of other approaches, you see, of genome editing and all that, you see. And I'm excited on the speed reading, you see, because one of the handicap that with even all these transgenics is that you have to, spend so much time in reading for creating homozygous lines, and also for having a sizable amount of seeds in order to hand over to the, for evaluation as a variety, you see, and then to do all the biosafety, to do all the biosafety work, you see. So with all this speed reading and all this genome editing and genomics, you see, there is an immense possibilities, and I'm happy to see that we have a similar facility being fabricated at NARC, and that is an excellent investment. And I'm sure that similar platforms could be built in Faisalabad and in Karachi also, and at other places, so that the breeders can utilize all that facility, you see. I, for one, am extremely happy to see all that, you see. Our own personal work, as you probably are aware, some of you are aware, that we have been working very actively on biofortification we had already developed wheat, transgenic wheat with increased bioavailability of iron and zinc by introducing phytase gene, which is exclusively expressed in the endosperm, you see. But all the other related information that has been given, I think that can also be very useful as far as the RN transport genes and other factors are concerned, you see. So I'm thankful to Shahid and Iqbal Chaudhry Saab for coming together and organizing this very important webinar. And we must continue to share this information, which will help in developing collaborations with all institutes who are working in these areas. And thank you so much for giving me this opportunity. Thank you.
Thank you, sir. Thank you very much. Your words mean a lot for us. We have Dr. Johar as well. Johar sir, can you hear me?
Hello, the Dr. Johar.
Johar is there?
Yes. We are unable to hear you, Dr. Sir. डॉक्टर साहब हाँ ठीक है Dr. Johar, we are unable to hear you.
So meanwhile, there is a question for Dr. Kavarpal. So if we can ask question.
Yes, I think that we can take few questions. Thank you, Awais.
So, Dr. Kavarpal, someone asked that you presented about the rate of the lower rate of genetic gain for wheat in Pakistan, and they have a question that what are the main reasons why we are little bit behind the global rate of genetic gain? There is almost 30% gap. So you work in CIMIT. So if you have a better insight on this.
Yeah, you might remember, I discussed that last year as well. You were in attendance. I think Shahid was there too. But anyway, I asked our star breeder, Ravi Prakash Singh. I suppose you know him, all of you, the wheat breeders at least. And 2 out of 3 varieties globally, they come from his program. I mean, that's the kind of impact that single breeder at the top has had globally on wheat production. And the first question I asked him was this, that why is genetic gain on a slow trajectory in Pakistan? And the first word that came out of his mouth was that there is the potential for genetic gain is still there in Pakistan. It's still going up. It's not stagnating, as I was made to believe back then. But it's slow because of slow varietal replacement. And I remember at the time, my recommendation, based on the Ravi's recommendation, was to use your extension department, boots on the ground, like I said at the time, to convince farmers to change their older varieties for the new ones. And according to Ravi, that alone could narrow that gap almost completely if you were to replace. And you told me that in the southern Punjab and northern Sindh, there are people who are growing 50-year-old varieties from the original green revolution back in the 60s. Yes, so that's those are the issues. I think those are the main things. And the other part, of course, this management and nutrient management and water is becoming scarce and so on. So there are so many of them. But the immediate cause that could be addressed is the varietal replacement.
Thank you very much. Dr. Johar, can we try to connect you and see if we can hear you? No, I think
he may have to reconnect. You should tell him to reconnect by chat if he can hear you. I had to do that once.
I think that, Wes, do we have any more questions?
No, we don't have any more questions.
I think I have one question to Professor Han.
Yes.
Professor Han, thank you very much for presentation. And, one question that I have is that, we have a buffalo chip, which is based on some of the genome, including Neeli Ravi from Pakistan. So at least for buffalo, I think that the available chip should be tried and, see how good are the data, probably we have to do a little bit more of GWAS and come up with the whether we can use the available chip. So do you have any comment on this?
I think yes, you're right, because we have been working together on the Pakistani buffaloes and also cattle genome resequencing of all the Pakistani breeds. I think, let's say today we have a really good understanding of the SNP present in each of these breeds. From there, I think we should look at it, because for the case of buffalo, there is already Afrometric chips, the 90K chip, which was built primarily with SNP from the river buffaloes. I think we could first, let's say we come up with our list of SNP. Probably we could have 5,000 our SNP of interest. Then we look at it, the inventory of the current buffalo SNP chip to see how much already included in this chip. If most of them, say 90% of our SNP are included in this in that chip, we could simply use that chip for genotyping. Otherwise, we have to consider to develop our SNP panel into a low density SNP chip to for genotyping. As what I mentioned, we are also struggling with this cost issue, because we cannot afford to say $15 per samples. That's really our problem. If we working on the cattle or the buffalo from Pakistan, the genotyping cost is still quite high. That's beyond, our budget. That's what the issue remained so far. I don't know really how we kind of go around, that's the really something, we have been talking, said this, the continuous sort of decrease of genotyping cost, but we have to think, say, for the commercial company, they have the sort of minimum threshold for a profit. If we push them down very low for per sample, then we have to supply really a very large number of samples. That's really the trade-off. I don't know how we go around. I don't really have a clear sort of mind, or I don't have a solution.
But I think that, thank you very much, Professor Han. But I know I've been talking to this breeder association. They said that, for at least for breeders, cost is not an issue. It may be a issue for small farmers, but for breeders, because they breed large number of animals, and the cost of a good buffalo or cow is very high. So as several 100,000 rupees. So I think that probably we can discuss this in further detail, that how we can use the available chips, and then especially in case of buffalo. So Wes, do you have anything to add?
Yeah, please check if Dr. Johar is back.
Yeah, I think, can you hear me now?
Yes. Finally, we can hear you.
Okay, that's great. So can I go ahead or?
Sure.
Yes, please. Go ahead.
Okay. Good evening or good morning to those who are on the other side of the globe. It was really a very inspiring workshop for me to hear about these new technologies from SNPs and editing to nutrition and malnutrition, and then hearing about the livestock side as well, which is my specialty. By looking at that, I think we would have to identify specific markers, QTLs or whatever you call it, for saline resistance, arid zones, and then for higher yielding specific markers for that. And then maybe we can go all out and go with, zinc finger, CRISPR, for oligonucleotide based directed cisgenesis, RNA dependent. We need to also get into methylation. I guess that's about time. So I'm always for, going all out. I don't want us to stop at one location. So speed breeding, grafting, non-GM on GMO, reverse breeding, agro infiltration. I think these are all the things, but one more thing I would like to add is translation and sustainability. That is our other problem as well. The cost that Dr. Han mentioned, I think we can add livestock breeding to the environmental issues, because we have negligible contribution into the greenhouse gases. So all the environment fund, we can add some for livestock breeding purpose, or maybe even biomarker discovery. Once we discover these markers, we can reduce the number of animals to a significant lower level. Right now, we have about 80 millions large ruminants, and then with all together, we have about 200 million ruminants at this point. So if we can reduce them, we can increase the efficiency. If we can increase productivity, we can also spare land for other crop production. Even by saying Dr. Tester, he mentioned that we don't need to increase cereal crops productivity, but in Pakistan, it's probably the other way around. We probably have to increase. We are still importing. By looking at the 2019 data, we exported $6 billion worth of agricultural product, even though we call ourselves agriculture country, but we imported $8 billion worth of items. While comparing Canada, they exported $62 billion worth of food items. So I think that's about time we need to go into, and I liked the point that I always wanted to sequence our animals all the way, because Dr. Shahid Mansoor is quite active in that area. There are other teams, but I would like not go into that. But what you mentioned, that we cannot use Holstein Friesian for Jersey, the same way I think we cannot use Jersey for our livestock. Sudanese livestock, if you compare that, because they have their markers identified by now, we are nowhere near that. So I think we have a great diversity. We have over 15 breeds. The diversity is the key to improvement. So in livestock sector, we are 1/6 of the productivity of the world. And by doubling it means we can drive our economy by 10%, the GDP rate, because we are contributing 11% to the GDP. So if we can double it, and doubling is, I'm sure, is quite easy. Even without breeding, we can do that. But with the breeding, if we bring that in, we train our farmers. Looking at Sri Lanka example, they just trained their farmers in nutrition of their livestock, and they doubled their productivity. But we have to go all out. We have to go for traditional breeding. We have to go for genetic marker-based breeding. We have to go for selection. We have to go for crossbreeding. We have to go for exotic breeds. So anything we need, I think this is time we have to take it on a war footage. We have to declare this as a national emergency, and we have to move into this area and start developing it. Most of our labor is in the poultry industry. Poultry is also something that we need to get into. Our backyard poultry is about 80 million, 84 million. and their productivity is 70 to 80x per head. So if we can develop that, if we can double that, that is another way of improving nutrition. So alluding into the nutrition side, just by doubling our backyard poultry productivity, we can achieve that goal. By looking at the bees population, we should probably not ignore that. We should not ignore our pollinators. We should not ignore our fisheries, because the biofloc is now coming in, and we should be going a step ahead now. We should be going into microbial side. We should look into the microbes. What microbes will increase our which crop productivity? Maybe we can start fermenting those. We can start adding those. We need to look into microbes to supplement our broiler diet. We need to look into microbes which can help us reduce the use of antibiotics in poultry. If we can reduce or eliminate antibiotics, we can easily earn and even export poultry meat. So I think I will say this was again a very eye-opening workshop. I would like to see some roadmap coming out of this. I'm sure Dr. Shahid Mansoor Sahib is very good at that. My seniors are there. I would like to see a roadmap, a tentative date, see what we want to do next. And I think we have to call it an emergency and move forward and implement these things, translate and sustainable. Thank you.
I think, Wes, do you have any announcement to make?
No, just there are a couple of questions. So someone asked if speed breeding can be applied to cotton. So I think there is no example yet, but NIPG is all set to test cotton in speed breeding. So I think they will have the results in 2 to 3 months.
Yes, I think so.
Last month I visited and they were planning for testing cotton under speed breeding.
Yeah.
So, I want to add one more point to my previous answer to you. I think you were looking for gene editing to improve genetic gain in Pakistan. We discussed that. Actually, that's quite. I'm not saying tongue in cheek. I'm serious. It's a possibility that if people are really they want to grow old varieties, we can directly introduce resistance into older varieties that are susceptible. Just want to bring it up. I didn't mention that. So now that we have the technology and we have enabled it in our lab, and I'll be happy to transfer it to your labs in Pakistan. It's not that big of a deal. So we can go that route as well. Sorry to interrupt, but I thought I should have.
Thank you, Dr. Kavarpal. I know that Awais has already provided you some of the lines. So
we haven't received them yet, so it's kind of like the MTAs and all that. We are still
but I think that we discussed about this, so we shortlisted some of the lines, like probably Inkalab and some of the major varieties that have been that have made a lot of influence in the country. So I must say, not only thanks to our speaker, but also our audience that they have been with us for almost 3 hours now. And I must say that this is a very good sign that we still have a lot of audience with us. And we are sorry that we were not able to admit all of the people who are interested, but I'm sure that that this the YouTube link will be available. And also I must thank Dr. Samar Yusuf and her team for providing excellent support, and LEJ for hosting this, and also all the other hosts like Comstech and Upsign. And thank you very much. Stay blessed. And I hope that we will come up with a series of, this was something desired by Dr. Kausar Malik, that we should come up with a series of smaller topics. It was just an kind of an update of what we have done 2 years ago. And I think that we will continue to have smaller topics and where we have more interaction with our students. Thank you very much. Stay blessed. Bye-bye.
Thank you.
Thank you.